Okay, so for this video, we're going to go over the anatomy of a typical motor neuron. This is kind of the structure of a neuron that you'd think of when you think of the word neuron. It's pretty much what you're always going to go over in a typical anatomy class. It's kind of like the intro to neuron anatomy. So this is what we're going to stick with. This is a multipolar neuron. There's also uni and bipolar neurons, but we're not going to cover them right now. So first of all, we have the cell body, which is right here. This is also called the perikaryon. That's like a technical name for it. This houses the nucleus as well as other cell organelles in the cytoplasm here. I didn't draw them for simplicity's sake. Branching off of the cell body are the dendrites right here. So each of these is a dendrite. And these little tiny projections coming off of each of the dendrites branches are called dendritic spines. And in anatomy, a lot of times we talk about the relationship between surface area and some kind of advantageous mechanism for the cell. So the dendritic spines definitely play a role in increasing the cell's surface area, and we'll talk about why that's important later. So coming off of the cell body is also an axon. So this whole purple structure here, this long tail-like projection, is called the axon. And the top of the axon right here is known as the axon hillock. And this kind of is wider at the top and then tapers down to form the rest of the axon here. The axon can also have like kind of a little side projection. This is known as the axon collateral. And this would just be like another kind of branch of the axon, and it would too lead to its own telodendromeres. But I didn't draw them just because it'd be a little crowded. So the axon I drew here is myelinated, which I drew with these orange blocks here. So this is known as the myelin sheath. The myelin sheath is made up of fatty cells and it's used as insulation for the neuron. In the peripheral nervous system, the cells that make up the myelin sheath are called Schwann cells. I'll write that here. And if you're talking about a neuron in the central nervous system, they'd be called oligodendrocytes. We're going to talk about Schwann cells in this video. We'll talk about oligodendrocytes in another. So if we cut the neuron like right here crosswise, we'd get something that looks like this. So this purple structure right here is the axon, cut crossways, and around it is the Schwann cell. So Schwann cells are long and thin, and they wrap around the neuron several times. And as it wraps, all of the cytoplasm is kind of pushed to the edge right here at one end. If you think about like a long balloon and fill it with a little bit of water, if you push it all to one end, it would kind of collect. And that's what happens here. It kind of bulges out at the very end, and that's where the nucleus and all the organelles would be located as well. This area is called the neurolemma. Okay, so that's the neurolemma there. And then the areas of the axon that are not covered in the myelin sheath are called the nodes of Around.I. And that's just the axon that isn't covered with the myelin sheath. Once we work our way down the axon, we end at the telodendria. So each of these little projections of the axon are known as telodendria. They have many branches off of them. Let me just label this here. So these are telodendria. Each one at the very end has what we call a synaptic knob. And the synaptic knob is eventually where neurotransmitters are going to exit the neuron into the space between two neurons known as the synapse. So there's kind of this space right here. So let's talk about how the structure of the neuron relates to how a signal is transmitted down. So as we talked about before, the dendritic spines give the top part of the neuron right here where the dendrites are a lot of surface area. So even though the entire neuron is able to be stimulated, it's most likely to occur here at the dendrites based on A, the amount of surface area, but B, the way the neurons are positioned, because you'll almost always see them positioned like this, where one neuron's dendrites are positioned very close to another neuron's telodendria. This is like a really simplistic way of showing it, but they're positioned in such a way where the dendrites are going to receive most of this stimuli. So dendrites are chemically gated, which means something such as a neurotransmitter can stimulate them. So up here we're going to say that they're chemically gated. Okay, so they're chemically gated, and this produces a graded potential, which we'll talk about in another video. So all of these graded potentials are going to stimulate most likely the dendrites, like we talked about. And this signal is going to be carried through the dendrites into the membrane of the cell body. So it could be here, carried through here, all over the neuron. So if the accumulation of all these different graded potentials all over the neuron are strong enough, an action potential will be formed in the axon hillock. So let's take a step back real quick. One difference between the axon and the dendrites is that, like I said, the dendrites are chemically gated. The axon hillock is voltage gated. So voltage gated, this means that it's going to form an action potential. Only if the all the graded potentials are strong enough to form an action potential. And we'll talk more about action potentials and the difference between graded potentials and action potentials in another video. But for right now, all you have to know is that if the graded potentials are strong enough, an action potential will be formed in the axon hillock right here. And then that action potential, it's an all or nothing signal. So if it's strong enough, it'll flow all the way through the axon to the very end of the neuron. So in myelinated neurons, like the one I drew here, the signal is conducted down the axon through saltatory conduction. So let me write that. So whereas a non-myelinated neuron has continuous conduction, which means it flows down the neuron in one just like even swoop, saltatory conduction means that the action potential is only carried on the parts of the axon that are not covered by the myelin sheaths, the nodes of Ranvier that we talked about earlier. So it can't pass through this myelinated portion of the axon. So it kind of jumps, so to speak, between these portions of the axon until it reaches the very end at the telodendria. So once it gets to the end of the axon, it's carried throughout the telodendria, like so, until it reaches the very end, known as these synaptic knobs. For this part, we're going to go to this little zoom in I drew here, which is a big close-up of a synaptic knob of a presynaptic neuron, which means where the signal starts, and then the postsynaptic neuron where it's going to be carried to. So the voltage signal that's carried throughout the entire neuron will end at the presynaptic knob, and this strong signal is going to open voltage-gated calcium channels inside the synaptic knob. And what that's going to do is going to open these channels, and calcium is going to flow into the space right here at the synaptic knob, and this flood of calcium manipulates these vesicles stored inside the synaptic knob, which hold the neurotransmitters, which are the chemical signals transmitted by neurons. So the calcium is going to change the vesicles in such a way that it moves to the edge of the synaptic knob and fuses its membrane with the membrane of the knob. As you can see, that kind of opens up the vesicle, and it allows the neurotransmitters that were held within it to flow out into the synapse. And the synapse is the space between the presynaptic and postsynaptic neuron here, and that's why it's labeled the synapse. So because of the close proximity of the synaptic knob of the presynaptic cell and the dendrite of the postsynaptic cell, these neurons, even though they're not touching, flow through this very small space and can actually fuse with receptors like this, specific receptors for each neurotransmitter on the dendrite of the postsynaptic cell. The neurons fusing with the receptors here on the postsynaptic cell is what starts the graded potential for the postsynaptic neurons. So at this point, we start all the way back up at the top, where we have these graded potentials caused by neurotransmitters here at the chemically gated dendrites. And then we're going to follow the whole process over again and continue on and so forth through a series of neurons.